Hello and welcome back to my channel, and it's very long time I'm uploading a video regarding molecular biology. uh, in the previous videos you have seen that molecular biology we have discussed, that is what is DNA, what is RNA, and how the replication, transcription, translation, and all these processes, post transcriptional, post translational modification has taken place. But the main part of the splice, that is transcription, is modification of RNA, because we already know that the transcription process is about formation of RNA from the DNA, because we use DNA as a template for the formation of RNA. We have already seen that process entirely, but now we are going to see very important part of this transcriptional post, you can say post transcriptional process, that is modification of RNA. You might have seen that how the structure of RNA is, but when the transcription gets over, or you can see when the transcription is done, the RNA which is formed is non-functional, you can say, or is full of certain junks or non-coding things which are not at all needed for the protein synthesis. And to make this RNA functional, to make the thing that is which is needed for the protein synthesis, we need to cut down certain things so that we get a perfect, or you can see, functional RNA. For that purpose, certain modification is done, and we have already seen many modification like 5' capping, 3' polyadenylation, and splicing. But in this video, I am going to discuss about specially about how the splicing machinery or the splicing mechanism takes place. And also I am going to introduce about what is the split genes and what are the splicing pathway, and it will be the part 1. And in this only I'm going to discuss only the first part of the that is the pathway, and the rest of the part will be coming in the upcoming video. So before going to this part, let's discuss that who discovered the split genes. So one of the two scientists, basically Sir Richard John Robert, and who was an English biochemist of the and molecular biologist, and Sir Philip Allen Sharp, who was an American geneticist and a molecular biologist, discovered this term, that is split gene. What is split gene? And we are going to discuss it in detail. So let's begin with our topic, that is split genes and the splicing pathway. Before going to split genes, let me make you understand what are split genes. So we have seen that the DNA segment is present. So DNA segment is basically divided into two parts. That is, one is intron, another one is exon. You must be wondering what is intron, what is exon. So you can see here, this is a structure of the DNA. Okay, this is a structure of DNA duplex, and these DNA duplex, you can see, is divided into many times, that is E exon, intron, exon, intron, exon, intron. These is none other than the split genes. This is called split gene. So why these are called split gene? By the name itself you can see that it is dividing, or it's splitting the DNA strand into number of small segments. That's why they are called as split genes. So don't get confused. If split gene is termed is used, it only means that it is being told about intron or exon. Okay, so why they are called split gene? Because they are basically what they are doing, they are dividing the DNA into small segments, or basically the splitting the DNA segment into small segments. What are introns? So intron is a segment or a section of DNA which is not involved in a protein synthesis. And what are exon? Exon are the section of DNA which is involved in protein synthesis. This is very basic definition of intron and exon. Let's get into deeper meaning. So for this modification of RNA, we need to do what we need to do. We need to just remove the intron, because these are the DNA structure segments of DNA which are not at all involved in protein synthesis. So what we will do by taking this intron? We have no use of it, and these introns are larger in size than that of the exon. So they are occupying much space in the DNA segment or in the RNA segment which is formed. So we need to remove that so that the RNA gets shorter and functional, so that only the functional part, that is the exon, will be remaining and protein synthesis can take place smoothly. For that purpose, the splicing mechanism is performed. So what is splicing? By this we come to the definition of splicing. So splicing is a mechanism or a pathway in which we are removing the introns and joining the exons so that we get the functional RNA. This is a basic definition of splicing. Next coming, what are the factor or what are the enzyme which are helping this splicing mechanism? These are known as spliceosome. So spliceosome is nothing but, spliceosome is the you can say spliceosome is the machinery, or you can say it is the it is a machinery that is helps in splicing out the intron. Okay, so the machinery or the genes which are involved in the splicing mechanism is known as spliceosome. By this you must be getting the clear concept about the basic terms, what is intron and exon. Now we going to move to the deeper part of it, that is how this machinery is performed, how the splicing pathway is performed. Okay, let's move to the next part. Before discussing the each one in detail, let me make you understand this splicing pathway mainly occurs in three parts. First is spliceosomal pathway, second is self-splicing pathway, and third is tRNA splicing pathway. In this part of the video, we are only going to focus about the spliceosomal pathway. Okay, so what is spliceosomal pathway and what is the mechanism followed, we will be discussing in detail. Let's begin. So in this spliceosomal pathway, what is involved? That is, a proteins is involved which is known as the small nucleotide RNA, or you can say snRNA. In this spliceosomal pathway, snRNAs are involved which helps in removing the introns. That's why these are known as spliceosomal pathway. So we are taking the help of extra proteins. Remember this, because this will be used in the upcoming video. You will be getting clear why I am using this term. Extra proteins, that is snRNA, is used to remove the intron. Whereas if you see in self-splicing pathway, as the name suggests, it is by itself only it performs the role of splicing, does not require the help of any other protein. And tRNA splicing is completely different from spliceosomal and self-splicing, which we will be discussing in the upcoming video. But for now we are going to focus only on the spliceosomal pathway. So before going to the deeper version or to the machinery of the spliceosomal pathway, let's understand the basic structure of the intron. So by now you must have understood what is intron, what is exon. Now what are the parts are included in the intron, which you need to understand better for the, so that you can get to know about the machinery clearly. So let's start. Wait a second. Okay. Before that, I am going to first focus on the exon. First see the exon part. So we can see that this is a 5' exon and this is the 3' exon. Sorry, the picture is cropped out, so that's why this is removed. So this is the 5, don't focus on this black written part, only focus on this highlighter. See, this is the exon, that is the 5' exon, and this is the 3' exon. Okay, 2 exons are present and in the middle the intron is present. In the first part only I have told that intron is larger than exon. This point you need to understand, you need to get. Okay, why I am saying so? Because you can see here that the intron is larger than that of the exon. We need to cut out this part of the segment of intron so that these two exons can join and form a short crisp structure which can be functional in nature. And what part of the introns are involved in the splicing process? Let's discuss one by one. So this is known as the 5' splice site. As I have seen here, I have shown you, this is known as the, this part is known as the 5' splice site, and this part is known as the 3' splice site. You can see here, this part is known as the 3' splice site. Then near the 5 prime splice site towards the upstream, you can see that GU segment is present, that is guanine and uracil segment, that is rich in guanine and uracil, and the 3 prime splice site is rich in adenine and guanine. Okay, to the upstream of this, okay, to the upstream of AG, polypyrimidine structures are present, and to that upstream adenine sequence is present, which is known as the branching point. So basically you can say for the splicing process, three things are required. That is, first is the branch point. Okay, what is the branch point? This A is known as the branch point. Second is, first is donor site. This 5 prime splice site is known as the donor site, and this 3 prime splice site is known as the acceptor site. So three things are involved in the process of what? Splicing. So this is the basic structure, or you can see the internal structure of the intron and the exon. If you understand this, then we can go to the machinery part. So let's begin. So first, what happened, as I have told you, in this spliceosomal pathway, first of all, what happened, this snRNA comes into play. What are snRNA? These U1, you can see here this U1, U2 and this U2AF, F65, all these are known as the snRNA, that is small nucleotide RNA. So first what happens, that is first an early complex is formed. So first segment you can see here, the first diagram, this is known as the early complex. What is early complex? So what happens, first the U1 will come and bind to the GU region, GU sequence. I have shown you in this in this diagram. This is the GU segment. So in this GU segment, the U1 snRNA will come and bind here. Okay. Then the splicing factor 1, that is BBP. BBP is the branch point binding protein, will come, and this is one of the splicing factor, will come and bind in the A site, that is the branching point site. A site is known as branching point site, as I have already told you. And then U2AF1 and U2AF2 will come and bind. So U2AF1 will come and bind to the 3 prime splice site, and U2AF2 will come and bind to the polypyrimidine site. That's why this figure is needed to be get understood properly, so that you can understand this complex. So first of all, this is known as the early complex. Okay. So first of all, this early complex is formed. Okay. That is, U1 will come and bind to this 5 prime splice GU region, and A region splicing factor will bind, and U2F1 will bind to the polypyrimidine structure, and U2F2 will bind to the 3 prime splice site. Up till now, complex E, or you can say early complex, is formed. Moving to the next part, what will happen here? Next, that is U2 snRNA will come and replace this BBP. It will replace the BBP and come and bind here. Okay, you can see here in this diagram that the U2 has replaced this BBP, that is splicing factor 1. Okay, and of course with the help of ATP. Okay, up till now this is clear. This is known as the pre-spliceosomal complex. Okay, pre-spliceosomal complex. Next, coming to the next part, next comes the trimer. What is mean by trimer? That is 3. Anything which is composed of 3 or 3 units or subunits is known as tri. Okay, so you can say that the trimer sRNA, that is U6, U4 and U5 will come. As soon as this U6, U5 and U4 will come and bind to this region, U2AF6 and 3,5 will removed. This region, which was binding to this polypyrimidine sequence, and 3 prime splice sequence will be removed. So what happens in this is known as the pre-catalytic spliceosome. So what happens, this U5, U4 and U6 will come and bind, and this U5 snRNP will come and bind to the 5 prime splice and U6 and U2. So this will form a complex which is known as the pre-catalytic complex. As soon as all of them will come and bind here, the U1 will be released. You can see here in this diagram, the U1 is released, and this total they form a structure. This is known as the complex B. Okay, this is known as complex B. Okay, now what happens, After this, what will happen? The complex C, that is catalytic spliceosome, will come and U4 will be released. You can see U4 will be released out. You can see here, next is the catalytic factor, that is U4 will be released. As soon as the U4 will be released, what it will do, the U6, remember it, listen it carefully, U6 what it will do, it will make a cut right here, that is in the 5' splice site of the intron. It will make a cut right here and form a lariat. Lariat means a hanging like region will be formed, which will join to that of the branch point, that is A. So now this part is free. Okay, so this part will be removed and will be joined to this branching point like a form of a lariat or a hanging region. Now what happens, next what will happen, the U5 will bind to the 3, then what will happen, this lariat will be formed and U5 binds exon at the 3' splice site and 5' splice site is cleaved, resulting in the formation of lariat, as we have seen. Next what will happen, the 3' splice site is again cleaved. Now first what happened, in the 5' splice site was cleaved, next what will happen, this 3' splice site will be cleaved. As a result, this whole total region is now free, that is the whole total intron is now removed. You can see here, this whole total intron is removed and the spliced mRNA is found, that is the 2 exon, that is 5' exon and 3' exon will be joined. Okay, you can see here in this diagram, that is 5' exon and 3' exon is joined and form the spliced mRNA. And this U2, U5, U6, all this snRNP are released, okay, and they are recycled. What do you mean by recycled? That is, they can be used for the another cycle, for another splicing they can be used. This all snRNP are recycled, that they can be used again, and the lariat which is formed or the intron is degraded. So the intron will be degraded and all the snRNP which is formed is recycled. So this is all about the spliceosomal pathway. So the major role which is taking part, or the main hero of this spliceosomal pathway, is the snRNP, because all the major activities is being performed by this. Okay, so that's why this is very important pathway. Usually this is a very confusing pathway and I have told the steps very precisely and one by one. You can pause the video and listen it to be very carefully again and again, because it's very easy. I have made you understood very stepwise and very easily. So if you have any doubt, then you can surely ask in the comment section. And this is all about the spliceosomal pathway. In the next upcoming class, we will be discussing about the splicing, self-splicing pathway and the rest of the pathway in the next upcoming video. So we will be doing this video at least thrice in a we will taking this YouTube classes thrice in a week. Okay, so that it will be coming alternative or consecutively 3 days continuously, or alternative days it will come. So if you have any doubt regarding this what I have taught you today, you can please comment and do like, share and share among your friends, because these are very important concepts regarding the modification, and these are usually asked in exams, and plus graduation also it is a part of your syllabus. You can share as much as you can among your friends, and thank you and have a nice day. And if you have any doubt that is regarding, you can say, molecular biology, what are this RNA, DNA, you will be facing certain problem, you can go back to the previous videos about what is DNA, RNA, what is transcription, translation, then you can come to this video for the better clarification. So that's all for today. Thank you and have a nice day.